The topic is the Disruptors, the next wave of promising psychiatry and neurology drugs. My name is Greg Harrison. I'm one of the SMID biotech analysts at BofA, and moderating with me is Dr. Maurizio Fava, Chair of Psychiatry at MGH. The panelists will introduce themselves as they discuss the questions. Maurizio, would you like to kick off Q&A for us?
Thank you, Greg. You've all worked on first-in-class treatments or very novel treatments that could have had a variety of indications. How did you select indication that you then pursued? How did you come up with that decision?
Sure. So in our case, we had developed a compound or combination of compounds that we believe targeted neuronal cell death. And that led to a choice. You know, there are many neuronal diseases where neuronal cell death may be one of the most important, if not the primary pathology. But I'd say a couple things happened. One, ALS particularly is probably one of the diseases of the most profound neuronal death, so it made a lot of sense scientifically. And I'll also say the ecosystem was just kind of in an incredible spot to become involved in ALS. We got to know many of the people from Mass General Brigham, as well as Dr. Maurizio and Dr. Rudy Tanzi, who really encouraged us and supported us as we kind of came into this field. So it was a combination of the science and the environment that I think made ALS particularly interesting for us.
So at Steve Davis, Acadia Pharmaceuticals, we have two marketed products. I'll touch on both of them from an indication selection perspective. Our first marketed product is Nuplazid, which is approved for Parkinson's disease psychosis. And there, the indication selection was based upon the mechanism of action of the molecule. So antipsychotics for decades have worked, they're kind of brothers, sisters, cousins of each other, and they work in a very similar way, and they all work by, in some manner, blocking dopamine. Our drug doesn't touch dopamine. It is a very selective blocker of serotonin. And by having a different mechanism, we have a very different tolerability profile. Most of the antipsychotics are burdened with high side effects. And our molecule has a very favorable safety and tolerability profile. So that made it ideal for elderly populations, such as Parkinson's disease psychosis. In addition, of course, if you're a Parkinson's patient, the last thing you want to do is block dopamine. That's the one thing you need more of, not less. And so by not blocking dopamine, we avoid the conflict that physicians and patients and families have between treating the motor symptoms of Parkinson's versus the psychotic symptoms. So that was kind of a natural fit. When we got approval in Parkinson's disease psychosis, we began looking at other indications as well. And working in an industry where their capital needs are great, the attrition rates are high, and the product development cycles are sometimes measured in decades, at least years. We took a shotgun approach and explored multiple indications at once, because once you have a molecule that has an appropriate safety and tolerability profile, you know the drug-drug interactions, you know everything about the molecule, you have a higher success rate, obviously, pursuing other indications. So we did that, and we're currently pursuing negative symptoms of schizophrenia. The reason we selected that is because negative symptoms, there's no drug approved to treat them. And so it was really medical considerations that drove that primarily. Our other drug on the market today is Debut. We just launched a couple of months ago for Rett syndrome. And that was truly a case where the indication was selected first, and the molecule, based on mechanism of action, was developed to approach that.
Sure, I'm happy to add in. So I'm the other co-CEO and co-founder of Amylyx, along with Josh. And just picking up on a point Steve made, you know, I think that this is what I hope is going to be a common theme in neurological diseases generally, which is I think there are approaches that are going to be genetically targeted, and I think there are going to be approaches that are broad. And I think that we've seen success in both cases, and often with combinations of those in other areas, probably particularly of note is oncology recently. And so I think for us, our first focus has been in ALS, but with our particular approach, we think that this is potentially a broad-acting approach, and therefore it may work in other neurodegenerative diseases as well. So we're working on initiating a study in a disease called progressive supranuclear palsy, or PSP. We have some early encouraging results in Alzheimer's as well. So I think that as we see the future, we hope that we can continue to develop treatments that act on broad or common pathophysiology, as well as more targeted approaches, as some of our colleagues on this panel are as well.
Great. So good afternoon, everybody. Ivana Magovčić-Libiš. I'm the CEO of Vigil Neuroscience. Vigil is a clinical stage microglia-focused therapeutics company that was actually formed by in-licensing 2 assets from Amgen. We actually have a TREM2 monoclonal antibody. It's an agonist, as well as first-in-class small molecule TREM2 agonist. So we believe in a precision-based approach to neurodegeneration, and what I mean by that, we really focus on indication where there's a very strong genetic, mechanistic, and biochemical association to microglia dysfunction. So how we chose actually our first indication was by looking at an indication that would check those boxes, and our first indication, which is now in a phase 2 clinical study, is in a rare neurodegenerative disease called ALSP. That stands for adult onset leukoencephalopathy with axonal spheroids and pigmented glia. It's a monogenic disease. It's caused by mutations in the CSF1R gene, which directly results in microglia dysfunction and reduction both in the numbers and function of microglia. And you would probably ask, well, why you're targeting TREM2. TREM2 is another receptor on microglia that's the main damage sensor in the brain, and CSF1R and TREM2 actually have downstream signaling that converges. So we actually believe that by overactivating TREM2, we can compensate for the deficiency in CSF1R, and there's direct genetic evidence actually in human genetic disease. And so we're very excited because we believe that that reduces the downstream translational risk and also allows us to get to that proof of concept very quickly. We chose to go with the antibody into a rare disease space and into these specific microgliopathies because these are diseases where the blood-brain barrier integrity is compromised in the areas of active disease. So we think the antibody as a modality is extremely well suited in that space. And then our small molecule, which is orally bioavailable, highly penetrant molecule, we think makes a lot more sense in larger indications, but again, taking that precision-based approach, we plan to go into the clinic at the end of this year into genetically defined subpopulations of Alzheimer's disease. Again, turning to genetics, looking at TREM2 variants where we know that the risk of Alzheimer's is significantly increased, as well as there's a more aggressive progression to the disease. So a precision-based approach, and that was how we decided on our first indication for both of the modalities.
Good afternoon, everyone. I'm Vimal Mehta. I'm one of the co-founders of BioXcel Therapeutics. I'd like to thank our moderators, Dr. Maurizio, as well as Greg, for giving us the opportunity to talk about BioXcel Therapeutics' story. We did not start with the indication. That's the question. We started with a combination of technology and the neuroscience. So when we started our program, our goal was that we use artificial intelligence platform to identify innovative medicines in neuroscience, and we focused primarily on the symptoms-based approach. When we looked at what are the symptoms where there is no treatment, one of the biggest healthcare burden was agitation. Agitation resulting from schizophrenia, bipolar 1 and 2 disease, as well as Alzheimer's disease. So we looked at and asked the machine and said, what can we find? And there has been no innovation over a decade in this space. Nothing has been approved. How can we make a difference in patients' life? Just to give you the magnitude of the problem, there are 140 million episodes that happen. And it's divided between schizophrenia and bipolar, about 40 million episodes, and in Alzheimer's related agitation, about 100 million episodes. So if we succeed, we can make a big societal impact. So that's how we chose our indication, agitation. And we were very fortunate that we were able to use our artificial intelligence platform to identify a drug now which has been approved. So we were able to go from first in human all the way to the approval in 3.5 years. I know this was amazing feat, combination of our technology to identify the underlying molecule and then execution by our drug discovery and development team. So now drug, as we speak, is already helping patients and we have launched the drug. Now we continue to expand this, its application into other indication. And as I said, Alzheimer's related agitation is a big one for us, and we are expecting pivotal data readout in June, so it's pretty soon. And also we are expanding the same molecule based on the data we have collected on thousands of patients to move into the major depressive disorder. So what AI and machine learning helped us identify was a drug which was already marketed, but it was forgotten and it was just sitting there, and we were able to take advantage of all the safety data and be able to direct using the AI and write a new chapter on this. So we are very excited that we are impacting the agitation space and then expanding it beyond into major depressive disorder.
Great. I'd like to ask the panel, when you're thinking about indications that take long periods of time to show a clinical benefit, such as Alzheimer's disease or others, how do you think about the right way to design a clinical trial to both increase your probability of success while generating approvable data?
Yeah, that's a great question. I'm making sure this is working. That's a great question. So I think in neuro broadly, we have a series of challenges that are somewhat unique. We can't biopsy the tissue, except maybe in some peripheral neuropathies, but in general, we can't biopsy the tissue. And our endpoints, in general, are quite variable. Often these are cognitive tests, motor tasks, that tend to be quite variable, both because of the nature of the test, but also because in many of these diseases, some patients may progress more quickly, while others progress more slowly. So as you said, this makes an inherent challenge that trials generally need to be large and long to have adequate statistics to detect an effect. I think the way we advance there is a fewfold. I think first, on the statistical side, there are more and more methods coming out that can help to reduce that noise. And those are so important, whether those are things like predictive models, things like different baseline characteristics or covariates that you can use to try to make a trial more precise when you're analyzing your endpoint. And also the endpoints are improving. I think we're seeing a ton of things such as digital biomarkers, at-home outcome measures coming to the forefront, which could, again, reduce this noise. But I do think it's a natural challenge of anything in neuroscience, the fact that you have to do large and long studies. And maybe the last point I'd make is, unfortunately, one of the solutions in neuroscience to the large and long studies is just to run large and long studies. I think that has been what's led to some of the recent drugs and some of the recent approvals.
Yeah, it's tough. As Josh mentioned, the brain's a very complex organ. Many of the diseases that we tackle, you can't look at a biomarker. It's not like measuring blood pressure. Particularly in the psychiatry space, you're looking at subjective endpoints. And if you're looking at disease-modifying therapies, which many times require longer studies to be able to measure disease progression, many times we're just have a conflict between wanting to make sure that we power the study appropriately and we run it long enough to be able to separate from placebo, but recognizing that the longer the period of time is that patients are on drug, that only lengthens the study by the treatment period, but more importantly, it just makes them more difficult to enroll. You have a lot more dropouts and it takes a lot longer to run a study. So sometimes we're faced with the unfortunate decision of, should I run it in a way that gives me the highest probability of success perhaps, but just limited by the practical reality that there's many times there are a limit in terms of how long you can dose patients. And so, and at Acadia, our priority always is probability of success. So we'll always sacrifice product life if we need to try to prioritize probability of success. So if we need to run studies longer, provided they're feasible, then we will. But the other strong consideration for us, at least, is in planning any clinical study, we get our commercial group involved very, very early because, well, the job number one, of course, is to get the drug approved and be able to benefit the patients with significant unmet needs. There are many things that you want to incorporate in your clinical design as well that may not necessarily be part of your primary endpoint, or your primary endpoint may not speak to. That'd be very important from a medical and therefore a commercial perspective. So we try to incorporate that early on as well.
Yeah, I really appreciate this question. I think it's one of the hardest things that we all do in the field of neuroscience. And I think just maybe two other things I'd add in. So one is the use of natural history. I see our longtime partner and collaborator, Alex Sherman here. Alex and some of his colleagues created a natural history database based on historical clinical trials in ALS. And now we're working on other methods to collect natural history. It is so essential because we have these questions about powering, about outcomes, about trajectories, about dropouts. We want to do trial simulations, which is an idea that can we model how things might work before we actually run the study. Well, those are great ideas, but if you don't have the data, if you don't have the database, if it's not workable, then you really can't test these things. So I think that we've seen recently just how important natural history is as we're designing these clinical studies. And I think we all can learn from some of the recent examples where I think this has been really meaningful. I think the other thing I'd add, which I think often gets forgotten in this whole process as well, is also to ask the people who have the disease and their families. I think there's so much that we can learn clinically and as we design our clinical trials as well. For our studies, we always have a patient and caregiver council to help weigh in. And it is just remarkable. I mean, I think we often forget that the people who are the most expert in a particular disease are the people who are living it every day. And you really change many of your considerations in clinical trial design based on the input you get from people who are living with it as well as their caregivers. So I don't think there's any perfect solution to how we do these things, but I think there are many different tools and things that we've learned from other fields that we can take now to neurological and psychiatric diseases to improve our chances of success.
Yeah, so I think I'm gonna say a lot of the same things have already been said. One of the main reasons that we chose the indication that we did, which is ALSP, is because there was availability of imaging and fluid biomarkers that we could actually study. But since we're the first ones into this indication, there's never been any work done previously, it was very clear to us that we needed to have a natural history study. So we actually started a natural history study, first of its kind, in this patient population way before we ever went into the clinical trials, exactly to Justin's point, to really start to learn about the patient journey, to really understand how these both biomarkers as well clinical endpoints that we were looking at are going to behave. And I can highlight the importance of really listening to the patient voice. It was critical in the designing of our natural history study as well as our phase 2 study that's ongoing now. So our phase 2 study, which is actually an open label study, obviously the primary outcome is safety and tolerability, but after that we're actually looking at efficacy biomarkers. So we're using MRI as and NFL as our efficacy biomarkers, and the confidence that we have that these are the right biomarkers is actually coming from our natural history study. And obviously we had this biomarker strategy from day one, but we're extremely encouraged to see how the FDA is looking today at biomarkers, how they're willing, once you can connect what you're seeing and the biomarker and the changes to functional outcomes, they're willing to consider biomarkers as surrogate endpoints. And I think that's really going to help us, get to the answers faster than we have been in the past.
This is a very interesting question, that how can you conduct the studies in Alzheimer's patients. So we are working on acute treatment of agitation. And agitation is one of the biggest reasons for Alzheimer's patient to end up going from their loved ones at home to assisted living facilities or to the nursing homes. And to be able to conduct a trial, there was a lot of innovation required, particularly in this patient population. So as I mentioned, we got our drug approved. It's called as IGALMI. It's being used for acute treatment of agitation, schizophrenia, and bipolar and 1 and 2 patients. But now we needed to work with the FDA, so we produced some data, went to the FDA, got a breakthrough therapy designation for the same drug that has been approved to work in the Alzheimer's related agitation. These are elderly patients, and the parameters are different, what you need to show in terms of the efficacy and safety for these patients. So as and when agitation event happens, CRO and our team, clinical team, has to work out an arrangement that you can go there, do all the measurements that are needed. It's episodic. It's not that it exists. You can enroll patients based on the previous history of their agitation episode, but when the agitation episode happens, you have to send the team. So we have to place CROs and our teams in those assisted living facilities or residential facilities where these patients reside. And when that episode happens, they will do all the measurements, pre-measurement, treated with the drug, and endpoint is 2 hours, so they will make those measurements. So it was quite a innovation that basically has to happen. And also there was no endpoint for measuring agitation in the Alzheimer's related patient. There is an endpoint. We got the drug approved in schizophrenia and bipolar. FDA was very comfortable. So with FDA, we had to work for the endpoint for the Alzheimer's related patient. So I would say innovation needs to happen at multiple levels. And if you're trying to develop a truly innovative medicine, it has to happen with the regulators. It has to happen with the key opinion leaders who support you, who has the experience of the patient. And then it needs to translate with the CROs who conduct the clinical trial and your own teams who has to see they have no previous experience of conducting this kind of a trial, and they have to design a trial. So it's quite a challenge, but at the same time, when you succeed, the reward is really big. And as I mentioned, that we have completed the phase 3 trial in 150 patients for treatment in the Alzheimer's related agitation, and we are focusing on ALF as well as residential setting, patients who have agitation episodes resulting from their underlying Alzheimer's disease, and episode frequency can be very different. So we are measuring all those parameters to be able to collect the data and then go to the agency. So it's very exciting time, and we believe these 100 million episodes that are happening, it can have a very transformative impact on the society, because all of us deal with it, and I personally take care of my mom, who is 93-year-old, and I see how the psychosis and agitation because of the underlying dementia can impact her as well as the family life. So we are very excited that innovation is happening, and there could be a potential for treatment for these kind of debilitating situations.
Over the years, I've noticed that Wall Street is always very happy when you get a new treatment approved, but within a couple of years, they're asking, what is next? In other words, what have you done for me lately? I'd like to ask the panelists, how do you deal with that pressure from Wall Street when you're really trying to get new treatments developed and kind of break new ground?
Yeah, well, first I'd say you're lucky to get a couple of years before they start asking that. But I think Wall Street is always looking for the next thing, the next progress, a reason to own the stock. Now it's approved, so what's next? And I think it's incumbent upon us as leaders in the biotech industry to keep innovating. I don't think you get a chance after you get a drug approved to rest on your laurels. I think that's the exact time to redouble your commitment in investment in new R&D, in investment in the space. And I think that's the only way to manage those expectations. I'd also say it takes education. Unfortunately, the timelines of biotech and the timelines of Wall Street are not the same. In biotech, a lot of the projects we take on could be a 2-year trial or a 7-year development program, whereas most investors in Wall Street are looking for quarterly or annual returns. And so I think that takes education, telling people often, frequently what the plan is, what the strategy is, what the milestones will be. And sometimes there can be that disalignment, but I think, again, what we have to do kind of as biotech leaders is keep pushing the innovation, keep pushing forward the things that can help patients. And ultimately, if we do well with that, our view is that it will ultimately all be fine with Wall Street as well.
Steve?
Well, it's a great question. I need to take a little bit of a running start of this. So what we've seen in the CNS space is, if we go back a couple of decades, the largest drugs in the industry were antipsychotics and antidepressants. And so they came out of the CNS arena. And as an industry, many times we invested a lot of capital to try to come up with a slightly improved version of drugs that already existed. That business changed due to regulatory changes as well as changes on the payer front, and that business went away. And that caused a heavy constraint of capital going into CNS. Many of the large pharma companies moved out, and the capital was then very judiciously allocated to products that truly are not just incremental, but very substantial improvements. So in our case at Acadia, pursuing a drug that didn't, as I mentioned earlier, didn't work through dopamine at all, and that was very unusual at the time. You see represented on this panel very novel approaches, and so the potential upside for these kinds of approaches is much more substantial. And it's been therapeutic, I think, for the industry, because we've seen now a renaissance in the CNS space, where now we're beginning to yield the benefits of this very focused capital investment, and with some really, really interesting new drugs. So you layer that dynamic environment that we all operate in on top of the comments I made earlier. We all operate in an industry where the capital investments are gigantic, the attrition rates are enormous, and the product development cycles are measured in years, if not decades. So that makes it very challenging to then pick the right opportunities or make the right investments to have a second drug or a third drug. The good news in CNS is it's so dramatically easier today than it was any time in the last, I've been in this industry almost 3 decades now, because of the focused capital investment in drugs that really are different. And so now what we're seeing, and we did a survey a few years ago to see where are VCs investing their capital. And they're doing it really in 2 areas in CNS. One is in psychiatric or broad indications, but those that are typically targeted for a subset type of population. Now, that doesn't necessarily mean niche markets. These sometimes can be very large markets, but a population that has an enhanced unmet need. And then, of course, in the rare disease space, and particularly in neurology, where we have a lot of probably single gene diseases, we're seeing just an explosion of activity there. So for us, and when I say us, I mean all of us on this panel, it's a function of trying to anticipate all the changes that I described on kind of a macro environment perspective, but most importantly, following the science. Follow the science, follow the data, and make investments accordingly. And if you do that, I'd love to say you just can ignore Wall Street. It's actually not practical to completely ignore them, but you can 80% ignore them and let the results speak for themselves.
Yeah, and I'll just continue on the same theme as my colleagues here. I think we're a little lucky in the sense that we're in the neurological psychiatric spaces where the research is just the most exciting it's ever been. And so I think there's this wonderful confluence where investors are very educated on these spaces as well. They're reading the same papers, they're watching the same clinical results. So I think that we can do this balancing act of focus on today, but also focus for the long term. And it's really tracking along with the science and medicine. And it's particularly exciting to get to say this at a conference like this with people like Dr. Fava and Dr. Tanzi, true world leaders in these spaces. And every single year there's something new that's coming out. So in terms of the what have you done for me lately, I think it's exactly as Steve was saying. If we stick to the science and we stick to the medicine, every single year we're seeing innovation. And so I think it's then our job as companies to try to think about, okay, how can we approach that? How can we take our platform? How can we take our insights and use what's being developed recently to develop new treatments? But I think it's a really, really exciting time in all of our spaces. So I think while it may have been more challenging in the past, I think if anything right now, it's about all of us keeping up with the advances as opposed to us being the ones to try to come up with it all on our own.
Yeah, I think we're certainly living in extremely exciting times today. For a long time, neurodegeneration was an area where it was not a place where investors wanted to go, right? It was a graveyard. But the recent successes have kind of encouraged investors that there is definitely opportunities in the space, so that's great to see people coming back into the space and being excited by the innovation. I think what's really important as a biotech company trying to engage with investors is making sure that you really have clarity in your strategy and in your value proposition, and that you continuously execute on established milestones that are clearly communicated to the investors, and then you will have the credibility, and that will allow to really keep them focused on what is the fundamentals here and where is really the value creation. And then it kind of goes beyond that, what can you do for me lately? It's really building that credibility with the community and executing on what you tell them you're going to execute. They're all very well educated, they're very well experienced, especially the investors who are biotech specialists. They know that not everything works, they know that failures happen, but what they really want to see is that you're going to do what you told them you're going to do, and that builds the credibility, and then they'll come back and invest with you again, even if something was not ultimately a success because the science did not work out.
I think I'd just like to build on that theme of credibility. I think that's so critical, and setting up the low expectations and delivering on those expectations is key. If you set very high expectations and not deliver, that can be very problematic. So I learned my lesson that we were able to get the drug approved in 3.5 years. Now you can be a victim of your own success because they could expect that you will do it again and again. So that sets the kind of. But it depends on the indication, it depends on what you are working on and how long does it take to get approval. Another thing I learned was that when we got approval from the FDA, there was nothing more exciting for the company. Everybody was elated. And here we come and we present that we got the drug approved, but stock goes down. And everybody is wondering why that happens. And we have proven ourselves to get the drug approved. We haven't proven ourselves as a commercial organization. So now our challenge is to build ourselves and prove to the street that we can commercialize this product. So we are in that phase now. So I think a lot of learning happens, but as everybody said, my colleagues on the panel, that it's building the credibility, setting the plan, showing the parameter that in the beginning, like you are heading in the right direction, this is the right kind of a matrix on commercialization or for your program and how you are expanding. And we always tell, we landed with our first indication and approval, now we continue to expand. And that's what Street is looking, that is there is sustainability in the pipeline or strategy that management is executing on. So it's always challenging, and we try to keep the balance internally, and one thing we have is create value for the patients. It will create value for all the stakeholders, whether they are investors or employees or anybody else. So we continue with that philosophy. Whatever we are doing, we are deploying the capital to create the value for the patients.
So given that the topic of this panel is the disruptors, and artificial intelligence is obviously a disruption we hear a lot about lately, how do you, members of the panel, think about the role of artificial intelligence when it comes to development within biotech and neurology specifically?
Yeah, despite being probably one of a younger person, I would say, you know, I might be a little bit of a dinosaur in terms of the adoption of AI. You know, I use ChatGPT. It you know, is pretty impressive what it can do. All that being said, I think as you use it for kind of more technical things, it is hard. I think the power of human expertise is still very strong. You know, so I think it will have a place. I think it's already being used in certain areas. For example, in toxicology, even before the big ChatGPT boom, there's a program called QSAR, and you know, Derek, which basically does a search to try to predict if compounds are going to be toxic. And I think we're going to see a lot more applications like that. For example, in dermatology or radiology, seeing that you know, a picture of a skin spot can be determined by AI or otherwise. I'd say for pure drug development, to me, it's still quite a team sport. Today, it's still done with a chemist, a toxicologist, a biologist, regulatory person, et cetera, et cetera. And I do think that while certain parts of it will probably be optimized by AI, I at least don't see in the incredibly near term, you know, us just asking an AI to make a drug and a drug comes out the other side.
Yeah, you know, I think part of it depends on how you define AI. I mean, we work in an industry, and particularly in the CNS space, where we do and have for years and decades a lot of computer modeling. So we do that when we, as Josh has mentioned, when we look at structural alerts for chemical compounds to see, well, there's certain things that increase the liability of side effects. We do it when we try to allometrically scale between results we see in animals on endpoints that where you can't ask the animal, are you depressed or are you having hallucinations, et cetera. We try to, you know, make those extrapolations into humans. And we do it, and we've been doing it for a few decades, just in terms of modeling out things that in an in silico model might be more likely to be effective against a particular target. So I think if there's an industry that's primed to benefit from, again, what we call AI now, but we've been doing a lot of this for a long time, I think we're it. So it's very exciting to think about the prospect of algorithms, models, advancing this even further. And again, I don't think there's a better opportunity to do that than in CNS, where you many times have these large populations, and you're trying to, we're always trying to predict the future in terms of what's this going to do if I move this nitrogen around the ring, or what's this going to do from a drug-drug interaction perspective, or what genetic profile of this is more likely to make this drug work or not. So I think as we go forward, we'll see more of what we've been doing, but hopefully at an accelerated rate.
Yeah, I'll go back to a comment I said earlier, where I think the uses of AI are only as good as the data that we have. And the hardest thing in all of the diseases that we work in is translation. What we see in the preclinic doesn't necessarily translate into the clinic. And I think the answer is we need better resources and databases from people with the disease, from biomarkers, from things that actually represent the clinic. So I think before we even get to the AI, I think we need the right data collection and databases in order to study it. Now, there are some really amazing resources. The one that instantly comes to mind is ADNI in Alzheimer's. I just think is a tremendous initiative. But I mean, that has taken hundreds of millions of dollars, enormous amounts of time, collaboration, operations, certainly a lot of federal government funding. So to me, the first thing is collecting the right data, and then we can apply the tools we have.
Yeah, I would agree with you, Justin. I think the quality of the data is critical in what we do. So having that in order to help us build, design better drugs, and I think AI can help us also potentially get to approvals faster by helping us really figure out which endpoints are going to make the most sense and can ultimately help us show that there is a change that we actually want to capture. But I agree. I think today it's really about the quality of the data, and if we can get an access to that data in order for us to be able to really design better drugs.
So I can speak on this topic for 40 minutes. I only have 30 seconds. So I will just say that AI is in our DNA. Even if you look at our NASDAQ symbol, it says BTAI. And without that, we will not have the drug approved. So we continue to use this technology. The only thing I will say is machine never get tired, humans get tired in reading the publication. Information is an overload. So I think we will need these tools, and we consider AI as an augmentative intelligence. We combine it with our drug discovery and development teams.
Great. Well, with that, I think we're about out of time, but I'd like to thank our panelists for joining us today, as well as Dr. Fava for moderating with me. Thank you.